You see my presentation, my file there too.
Yes,
All
we
right,
can see your presentation. Yes.
Okay. I'm going to be talking about Phytophthora. This is a plant pathology presentation. If you are not a plant pathologist, the science of plant pathology started by diagnosing Phytophthora problem in Europe in 1845-46. Was late blight of potato caused by Phytophthora that time. That time we had only one species of Phytophthora. Right now we do have more than 150 species of Phytophthora in the world. One of them is Phytophthora capsici, or you may call Phytophthora capsici. Phytophthora capsici was identified exactly 100 years ago in the United States, in the states of New Mexico, on pepper. Right now, we do have this pathogen all over the world on more than 50 different species. So any question later on might come, you can send to my email, babadoost@illinois.edu. So why this is important? Because Phytophthora capsici is right now is worldwide. It attacks to 50 species of plants, mainly crops, and all over the most important host crops are pepper and cucurbits. Cucurbits like pumpkin, squash, cucumber, melon, watermelon. So I'm going to be focusing today, basically, how this disease is on peppers and cucurbits. Those are the main hosts in the world. I'm in Illinois. If you want to know, I'm roughly about 200 kilometers south of Chicago at the University of Illinois. This is the largest university of Illinois. And as you see on pepper, first on pepper, we can see the disease from the time we put the transplant in the soil. Here it is. See the lesion in the crown section at the soil level. Then the pathogen can attack and infect the plant anytime from the beginning up to the harvest. You see, these plants are dying because of Phytophthora. This is a well-developed plant and infection here. And particularly infection in pepper start from the crown section and the root, then moves up. You see, this is chili pepper, and this is a pepper field. There was about 200 acres in Illinois, some about 20 years ago. Then after the rain, and the plants were setting fruit, everything died. This is so nasty, so aggressive pathogen. But not only the roots and the crown section are infected, but also fruits are infected. You can see the fruits. This is the Phytophthora. By the way, this is very easy to work with this pathogen because we can easily culture this pathogen and we can inoculate plant either in the greenhouse or in the field, get all sorts of data. Is a nasty pathogen, and every cycle could be repeated in about 5 days, 7 days. Multi-cycle pathogen. So when it comes cucurbits, in cucurbits, there is no root infection. Mainly infection is at the soil level or above. You see up here, seedling death. Similarly up here is in the lower part of the plant infected and whole plant dying. Here you see the vine is infected. Here you see the fruits are infected. Just it goes very fast. All the pathogen needs moisture and also warm temperature. When I say warm temperature, above 20 degrees, below 30 degrees. That's the perfect. You see, cucumber, melon, watermelon, pumpkin. And this, for example, in pumpkin, this lesion can develop in about 2-3 days. And you can see millions of sporangia on the fruits. They can easily detach and spread. So this is an oomycete pathogen. You know, if you don't know, oomycete used to be as a fungi, but no longer true fungi. We call them fungal-like organisms. Genetic and the pathogenic variation is enormous on this pathogen. For example, I will show you, we have broad genetic and pathogenic variation among the isolates from Illinois. Multi-cycle disease survives mainly as a oospores and also mycelium. Does not survive too long, with mycelium mainly survives as an oospore. Mycelium in about 1 week, not 1 year will go away, but oospore survives about 4 years in the soil. Favorable condition, moist and warm condition. I am talking about variation among isolates. For example, in Illinois, we isolated 4 different morphology differences of the Phytophthora from cucurbits and the peppers. Here they are 4 of them. 4 of them. All of them produce sporangia. Sporangia produce zoospore. Zoospores just to move. When they move, that's called a zoospore, comes from animal. And oospores, in the bottom, you see oospores are pretty thick-walled spores. They are very resilient. You see this one, genetic distance, a genetic variation. In Illinois, depends where you go. For example, this is from Illinois. In Illinois, we detected 6 different groups based on wrapped group. All 6 groups are a little different, as you see in the tree. And genetically, they are different. And also, they are responding to fungicide differently. So if somebody working with this pathogen any place in the world, I know, for example, this pathogen is very important in India. It is important in China. It is important in Africa. It is important in Middle East. It is important somewhat in Mediterranean area where you are located. So in any place, when we're working with this pathogen, we cannot just rely on the research on one isolate. We have to see how many isolates we have. For example, if we want to develop a resistant varieties of pepper, we have to test all possible isolates because genetic differences. So if somebody comes and tells me, okay, I do have this pepper accession or pepper cultivar, it is resistant to Phytophthora capsici. I would ask, did you test it against the isolate from Illinois. If not, it may not be resistant to isolate to Illinois. So this is very, very important all over the world. Host strains of this pathogen, we do have, for example, we do have pepper and cucurbits. And we do have other hosts, but they are not as important as peppers or cucurbits are. For example, we do have this pathogen on tomato, but it is not really very serious. So we have tried to really determine the host range of this pathogen in Illinois to make sure that we know what are the hosts, what are not the hosts. For example, here in vegetable, in the crop, we do have 36 different crops, and we do have 9 weeds in Illinois, mainly planted in Illinois. And we determine what are hosts, what are not the hosts. This is very important for crop rotation. If we're going to say, okay, make crop rotation for 2 years, 3 years, 4 years, what you have to consider in your crop rotation. The host, for example, cantaloupe is melon, is host. Cucumber is host. Gourd is cucurbit, host. Eggplant is host. Pepper is host. Beet is host. Pumpkin, squash, radish is host. Zucchini, watermelon, turnip, honeydew, Swiss chard are host. In comparison, I'm not going to all of them because a slide will be available for everybody. You can look at. For example, corn is not host. Pigweed is a weed, is not host. Soybean is not host. Broccoli is not host. You can see radish is host, but broccoli is not host. Kale is not host. Cabbage is not host. Crabgrass is not host. Basil is not host. So chive is not host. So you can see right now, everybody in Illinois knows if they want to do crop rotation to minimize the inoculum buildup in the soil. This is a soil-borne fungus, soil-borne pathogen, I would say. And what should consider for crop rotation. By the way, my presentation is pretty much problem solving. So not only we determine the host range, but also we determine survival of the pathogen in the soil. For this purpose, we just produce lots of oospore. Oospore is surviving body of the pathogen in the soil. Then basically we have 4 different soil types, and we collected 4 different soil types and then used 3 depths, 2 centimeter, 10 centimeter, 20 centimeter for 4 years. You can see here the experimental side. We used PVC tubes because we want to put the oospores in the soil, in the bags, in just a water penetrating, water moving a bag, and put in the PVC tubes to be able to remove. And then removed every 6 months for replication. Every 6 months, what we found, when we put the oospores in the soil, they just go down quickly, but not eradicate. Even after 48 months, we were able to separate some oospores from the infested soil, meaning that you don't have to have millions of oospores in the soil. Few oospores can initiate disease, then the pathogen will produce sporangial zoospore, then build up quickly. So even 48 months later, we were able to separate, to extract sporangia and to extract oospore from the soil. What about viability? Viability declines too. We found oospores are viable, meaning that we can culture them, we can inoculate plant, they produce disease. After 3 years, they were viable. But after 4 years, they did not produce disease, no longer viable. What is the importance of this finding? 4 years of crop rotation with non-host and controlling the weeds seriously would be adequate to get rid of this pathogen in the soil. You know, we don't have to really struggle. We know the epidemiologically how this pathogen behaves. So pretty much this was good for crop rotation, which is very important for management of this disease. Then we checked for resistance, any cultivar resistance. We found resistant cultivar of peppers in Illinois, and they are available in other parts of the world. But there is no resistance whatsoever in cucurbits. There is no resistant cultivar in India. There is nothing in Mediterranean area. There is nothing in Australia, nothing in the United States, nothing in Latin America. This is a huge problem in Latin America. So this makes really complicated for cucurbit problem. I'm going to go through for each one how we take care of. For really determining a resistance in pepper, we can easily grow pepper. About 4, 5, 6 weeks old pepper transplant, inoculated in the greenhouse, and then you can see the lesion develop in maximum 2 weeks. In about 1 week, 10 days, you can see the lesion, and the plant dies. Defoliation and then dies. And you can see we were screening a huge number of the cultivars in accession in the greenhouse. You can see, for example, this is perfect. This is all gone. Very easy to work. And one of the easiest pathogen I have found so far to be worked in the greenhouse and in the lab, even in the field, to get the result. But greenhouse is complementary information. We have to go to the field. There is the field. We screen in the field, infested soil in the field. You can see what cultivar is okay, what is completely dying. Some of them dying quickly, some of them dying a little bit later. If you want to inoculate peppers in the field, you can do that too. Combining the greenhouse information with the field information, with the all isolate from the area, at least from the states, will give you really very reliable information on selecting resistant varieties. Based on selection in Illinois, these are resistant cultivars varieties of peppers. For example, Alliance, Aristotle, emerald isle, enza, intruder, paladin. Paladin is one of the pepper cultivars is resistant not only in Illinois, but I know most places in the world. Pretty good. And the Ranger and the Revolution. These are very good. Those in the yellow sign, intruder and the paladin, highly resistant, meaning that about 90% resistant. Those, for example, in the white color, they are some resistant. For example, 60%, 70%, that's so resistant. But some growers do not like a paladin, do not like this resistance. They go whatever market demands. For example, if they plant paladin, they may not be able to market easily. They have to grow whatever the consumer really wants. So this is good. This is one way. If somebody wants plant paladin, is fine. Plant revolution is fine. But if does not, we have to offer something else. What is something else? We have to offer fungicide problems. Fungicide for controlling this pathogen. We do have in about 2000, 1999, 2000, 2001, we had almost no really fungicide taking care of this pathogen. Not in Illinois, not other parts of the United States, other parts of world either. But in the past, I would say 22 years, we tested more than 50 potential fungicides, and some of them are pretty good. For example, here I listed those are pretty good in Illinois and more or less in the United States. These are common name, these are commercial name in the United States. This commercial name might be different, for example, in China or Australia, or let's say in Turkey. Cyazophamet runman, for example. And you can look at it. We can look at it. One of the compounds just came available a few years ago, just 4 or 5 years ago, oxatiopyroline, horrendous. This is very new and very effective. And also, those plant pathologists know that we have to consider FRAC code. FRAC stands for fungicide resistant action committee. For example, we cannot use whole season fungicide from the same group. We have to alternate with different FRAC groups. You see this one, we have FRAC groups like 21, 40, combination 40, 45, 22, 11, 27, so on. So we do have right now good combination of the fungicide to take care of this pathogen in both peppers and also in cucurbits. For Illinois, these are most effective. Horrendous alter, FRAC code of 40, 49, Revis, FRAC code of 40, Runman, FRAC code of 21, and we mix it with the silver, L77. This is spreader sticker. Illumine, FRAC code 22. Presidio, FRAC code 43. These are what we recommend in Illinois on peppers and also for cucurbits. Very, very good. However, I would say these are not 100% guaranteed, because if we do have a rain, sometimes in Illinois, we do have 5 inches rain right away. 5 inches rain means that, what, 12.5 centimeter, 125 millimeter in about a couple hours or 3 hours or half a day. Nothing really effective in this case. So we have to spray the plants before the rain and after the rain. If water is standing in the soil, it's going to be very, very difficult. Another important point is we have to spray the plant at weekly schedule. If we don't do at weekly schedule, they may not be really effective. We have tested at 10 days interval. For 10 days interval, they were not as effective at 7 days interval. And always we have to follow label direction. That's important, to follow label direction. So what is another approach we took was red light management. We were so excited. What is the red light management of pepper? Here it is. We grew seedlings of pepper under red light. Red light is 650 to 700 nanometer wavelengths. We do have tubes available. Red light is pretty much used in Japan too. And then when we grow under red light, these are in the left side, grown under natural light. And these are grown under red light. They grow really faster and plants are pretty strong. Then when we inoculated all of those grown under natural light, but those grown under red light, they become resistant. Not 100% resistant, about 80% resistant. We were so excited that we were going to be solving the solution of this pathogen in Illinois. However, when we exposed the plant to red light for 4 weeks and planted in the nature, in the field, gradually they lost resistance. So at this moment, we are not using red light treated plants in the field. If anybody has this Phytophthora problem in indoor production, like a high tunnel, like a greenhouse, and they can use red light, they would be good enough, very, very effective. But if you have transplant and then take it to the field, you should not expect full season resistance of the plant. So what is our recommendation for pepper at this time in Illinois? I'm talking in Illinois. I'm not talking about New Mexico. I'm not talking about North Carolina. This is in Illinois. Plant-resistant cultivar. We do have available if you want. If you don't like, do 3 years or longer, rather 4 years of crop rotation with a good weed control. Grow on the raised bed. Grow on the raised bed to minimize the moisture effect, because raised bed will be a little bit above and there will be less moisture. Avoid using contaminated water. If somebody has a pond, small lake, and there was runoff water from infested soil to the pond water is contaminated. If somebody is using the water from that pond, then spreading the pathogen all over the field. Do not do that. Fungicide application, again, at 7 days interval. You can use combination of tolerant cultivar and some fungicide application. This is a good way of really managing this disease. I don't use control. I'm saying managing this disease. Never we really accomplish 100%. If we accomplish about 95%, 90%, this is great. Then when we come cucurbit, that's really complicated. We have to use integrated management approaches. Why? Because we do not have resistant cultivar. When I'm talking about cucumber, pumpkin, squash, melon, watermelon, and so on. None. No one has resistant cultivar, resistant varieties against Phytophthora capsici. If anyone in the world comes up with a resistant cultivars of any cucurbit against this disease, will be hero. Can easily publish the paper in any journals. We don't have it. So what we have to do it, we go step by step, integrated management. 3 years, rather 4 years of crop rotation. Seed treatment. We developed the seed treatment with Epron XLLS. Epron XLLS is a commercial name. This is mefenoxam. The common name is mefenoxam. How much you need? Almost nothing. Almost no cost. 0.4 milliliter of this compound. Apply to 1 kilogram of the seeds and plant it. That's good enough. That takes care for 5 weeks. I'm going to show another slide. Avoid using contaminated water. Disc localized infected plants. I'm going to show a slide. Fungicide spray is essential. Sanitation is essential. I'm going to show some related slide. For example, seed treatment. Seed treatment here with the Epron XLLS mefenoxam. I put this one in the US system, English system, 0.64 flow ounces per 100 pounds of seed. That's exactly what I mentioned for 1 kilogram of seed. This is great. When you do this one, you protect your plant for 5 weeks, not beyond 5 weeks. Prior to the seed treatment, we have total losses of the seedlings in the field, in pumpkin and squash. Ever since we're using this one, almost nothing. Perfect. Then we go to disc. Disc localized infected area. When we do have a large field, for example, in Illinois, this is a pumpkin field. In Illinois, 200 acres, let's say 200 acres, roughly about 80 hectares. And usually pathogens start in the low area. We have to go really check the field every bit. In the low area, particularly after rainfall, if the disease started, we have to get rid of those plants infected. This way, we prevent inoculum building up. But getting rid of few plants would be good enough. In a small area, like 1 hectare, you may just pull out the plant and take away. That will prevent building up the inoculum. Then we're talking about fungicide application. Fungicide application are essential for vine infection and fruit infection. Believe me, we had 100% of losses in pumpkins and squash prior to fungicide application. I mean, we could walk in Illinois, field after field, and see 100% of crop losses. So, but right now we don't. Rarely we have about 10% in very wet conditions. We have to apply the fungicide. Those fungicide is very effective in Illinois at weekly schedule. What are those in Illinois? Orendus ultra, Revis, Runman, Elome, and Presidio. These are most effective ones in Illinois. I do have, every year, I do have experiment, trial, make sure that everything is under control. I have been doing a trial for 23 years. And I go hotspot. I go hotspot where the disease is the highest. Then growers can see, and the information, the data we collect are very reliable. We have been very, very successful, frankly. So what is the overall recommendation? Here, again, we go to list, but at this point, I'm going to go sanitation. This is pumpkins. Pumpkins are very popular in here. China is the biggest pumpkin growers in the world. India is very big in pumpkin and particularly in squash. I guess United States is number 4, probably. Illinois is the leading in pumpkin production. Since this pathogen is in the soil, usually it comes in the bottom. Most times infections start in the bottom. And occasionally we don't see the infection in the bottom. One day, one of the growers in another state contacted me, said, he's just harvesting a pumpkin, taking it, healthy pumpkin, healthy looking pumpkin, taking to the market after about 1 week, all of them comes back infected. When I visited, I realized the harvester, the person is picking up the pumpkin, is just taking from the stem and the left hand, let's say left hand in the bottom, then realize this pumpkin is infected, then drops. When drops, his or her left hand is contaminated, then goes and picks up an infected, healthy pumpkin, then practically spreading the pathogen from hand to the healthy pumpkin. Then they put all in a bean. Bean is big container. Then take to the market, and then warm condition and moist condition, all of the healthy pumpkins get infected. So if somebody is picking up, let's say watermelon or pumpkin or squash, winter squash, if hands gets contaminated, he or she has to wash hand with soap truly or use alcohol, decontaminate hand. If you harvesting pumpkins or harvesting watermelon or squash or anything, cucumber, do not put in the field which has history of Phytophthora blight, because soil is contaminated and the fruits will die. With this one, thank you so much for your attention.